Yeah, thanks for the introduction. And also, I really want to thank you for welcoming me and my other colleagues at Henry Ford into your department. Really, the way you've reached out to us and connected many of us has been really, really amazing. And I think the process has revealed a lot of similarities in the culture between our two departments, which I think has already fostered a lot of goodwill and scientific collaboration. So, yeah,
absolutely agree.
Yep.
I think everybody who is not Al, I'm going to ask to mute because I think I'm getting a little echo. It could be me and Al causing the echo, but I'm going to turn mine off and hopefully we'll get better. Thanks,
Yep.
Al.
Thanks, Don. So yeah, my name's Albert Levin. Most people call me Al. As a quantitative person, scientist, I really have a lot of love for underutilized data, where we can apply new methodology to ask clinically relevant questions. And this talk is really an example of this sort of kind of secondary data analysis from, an initial African-American prostate cancer cohort study, retrospective cohort study that we did. And yeah, kind of a cool application of a new area that we're getting into of analysis of image analysis from histopathologic slides. So, I mean, most of you probably don't know me. I just wanted to give you a little bit more of an introduction into who I am. I'm a genetic and molecular epidemiologist by training. I've done the majority of that training at the University of Michigan as a master's student in epidemiology. I should also say I have a master's degree in statistics as well. But as a master's student in epidemiology, I did work with scientists at the University of Pennsylvania studying prophylactic oophorectomy as a means to reduce breast and ovarian cancer risk in BRCA1 and BRCA2 mutation carriers. Then during my PhD, which seems like a really long time ago, it was when the microarray technology was first coming online and we could do transcriptome-wide analyses. And I was involved in a cancer study with really the first generation arrays that was sponsored by the NCI Directors Challenge, they called it at the time. And it was a really exciting time for methods development because before everybody was focused on single genes, and now you could analyze these, ohm-wide data sets. And so there were a huge explosion of methods, and I developed one to analyze transcriptome data in the context of the chromosomal context. And then as a postdoc, I got more into germline genetics and studying the germline genetic risk of prostate cancer. In 2008, I joined the Department of Public Health Sciences at Henry Ford, and I was really introduced by a colleague of mine, Dr. Ben Rubicki, our vice chair of epidemiology within the department. He really introduced me to this field of racial disparities, disparities in general, but racial disparities of disease risk and severity in general. And that is a theme that really pervades a lot of the work that we do at Henry Ford and pretty much all of my research. So I kind of have three areas that I focus on, so not very focused, but so I study the genetics and environmental determinants of sarcoidosis risk in African Americans. All these conditions that I'm in the list are ones that are have multifold higher risk in African Americans relative to European Americans. And then I also studied the genetics and pharmacogenetics of asthma and allergy in African Americans, both in kind of individual and consortium-wide studies. And then the work that I'm going to do today comes out of my study of the molecular epidemiology of prostate cancer in African Americans, and specifically from a DoD study, a study that was funded by the Department of Defense, where the primary focus was to study integrated DNA-based biomarkers, both somatic copy number alterations and methylation changes within tumors to identify aggressive disease in African Americans with prostate cancer, and specifically for African Americans, again, because they're at increased risk of developing prostate cancer, increased risk of more severe aggressive disease, and that, there are very few molecular studies. The majority of the molecular studies of prostate cancer are really focused on majority populations like Europeans or European Americans. So I also, as Dr. Misra said, I'm the vice chair for the Division of Biomedical Informatics, and we have 6 PhD faculty. There are 2 who are coming online relatively soon. I just padded my numbers to, include those 2. And then we have 5 master's level analysts. And just as a shameless plug, we're hiring now. So if you have any interest, I think I talked with Gustavo, and I think there was circulation, but if you want to, you can contact me if you're interested. And so we have 3 subgroups within our biomedical informatics division. The first is kind of where we started, which is really in the genetic molecular data analysis area. And this is, done in bulk tissue and all various forms of single cell varieties, spatial transcriptomics, and all manner of omics data. Our second group is medical informatics team, where we're focused on data mining of the electronic health record, which at Henry Ford goes back into the 80s. And a lot of this work is focused on developing clinical support tools for physicians, nurses, and other healthcare practitioners to streamline treatment, improve efficiencies and outcomes for patients. So we're highly integrated with operations there. And then the third group is really reflective of what I'm going to talk about today, which is our medical and histopathologic imaging data analysis group. And that really grew out of a mentoring relationship that I had with Dr. Yale Chen, who's since left the group. But he really introduced me to this field of digital histopathologic image analysis. And a lot of the work that I'm going to present today is work that he did in collaboration with me. So this area of histopathologic image analysis is a developing one, and developing very rapidly. Is indicative of the field developing. It doesn't really have a good name yet. Some people call it quantitative histomorphometry, some histomics, pathomics, digital pathology, but really it's the use of computational methods to extract and analyze data from digitized histopathology images to generate results and tools to further refine disease diagnosis, prognosis, and as well as prediction. And it has a lot of significance in cancer research in particular, although it's not just germane to cancer, really applicable to any application or condition where there's surgical samples, tissue samples. So it's being used as, for early diagnosis of aggressive cancer, really to aid in tumor aggressiveness classification based on existing paradigms, which is something that that's really what I'm going to talk about today. The work that we're doing is really within the existing paradigm and just trying to augment it. And this is about Gleason grading in prostate cancer. Which is Gleason grading is reflective of the degree of differentiation of a particular tumor, which is directly linked to how aggressive it is and related to recurrence. And then it's also revealing previously underappreciated biology that could be used to change the existing pathological classification paradigm. So there are lots of examples. Oh, wait a minute. Oh, sorry. Yeah. So just, what are histopathologic images? There are high resolution scans of tissue specimens obtained for the purposes of diagnosis and treatment planning. These are surgical specimens. They could be biopsy based. They could be surgical resection of, part or all of an organ. The specimens are cut onto microscope slides. and they're thin cuts of about 5 microns. Most are, assessed by a pathologist after hematoxylin and eosin staining, which is used to stain the nuclei and the DNA and the cytoplasm, respectively. And then these slides can be scanned at high resolution, typically between 20x and 40x for additional analysis. And like I said, the H&E is really the most common staining technique used for diagnosis, and there are millions of these slides. So again, I talked about, loving underappreciated data. I mean, again, it's sort of any condition that has surgical tissue and generating these. There's definitely additional things to be learned from them. Did I? Oh, okay. Sorry. So there are lots and lots of examples of the application of histopathologic image analysis in the cancer literature, and I'm just going to go through a couple here. The first is from using data from the Cancer Genome Atlas, or TCGA, and specifically in this case, the non-small cell lung cancer portion of it. The figure on the left is an example of model construction, classification using multiple different models, bagging, naive Bayes, random forests, support vector machines with different kernels, to use the histopathologic features to differentiate the tumor, non-small cell lung cancer tumor versus benign tissue. And on the right, they're using those features extracted to build a classifier of overall survival. This second example is also based on data from TCGA, and it utilized H&E images to identify and quantify tumor infiltrating lymphocytes from 13 different cancers in about 4,800 individuals. And this was done with very high fidelity and based on, in comparison to the human pathological assessment of the TILs, as well as a molecular assessment of the TILs. And this is really important because immune infiltration is an important predictor of cancer outcomes, or can be an important predictor of cancer outcomes, and also who should or should not be assessed as a candidate for immunotherapy. So histologic changes in tumors can also reflect molecular changes. And this manuscript that I'm putting up here details how OncotypeDx categories of breast cancer are predicted based on digitally assessed histologic changes. And so OncotypeDx is a 21 gene expression signature that splits estrogen receptor positive breast tumors into high and low risk categories of recurrence. And so using multiple methods, the authors showed that a support vector machine approach based on digitally assessed histologic features could classify these OncotypeDx categories, these molecular categories, with a high area under the receiver operator characteristic curve. The other thing that I wanted to point out here is this is also a nice example of how these analyses are done. So typically, so if you look at the image, you can see all these boxes laid out as a grid over the tissue, and that's typically that's an approach that's typically utilized, and one we utilized in the data that I'm going to show, where you break down the bigger image into smaller square tiles, and those serve as the unit of inference. And then the final classification is based on a function of those individual tile predictions. So here you can see that these the green ones are predicted to be low risk OncotypeDx. And then the blue ones are the high categories. This last example that I'll show is one where again from TCGA and the brain cancer or gliomas, looking at recurrence risk prediction. And so this approach used a deep convolution neural network approach to do feature extraction from the images, and also integrating genomic data with that, with the histologic characteristic information. And then they layered on a terminal layer with a Cox link function to predict overall recurrence risk. And what they found was that the best prediction was when they fused together both the histomic and the genomic data. And they were also able to map some of the deep features that were very important in the prediction of recurrence back to areas of the tumor or adjacent regions, actually, where they could see that that one of the components of the histology that was predicting survival was this edema in adjacent benign areas, so outside of the tumor region. So I've used this term feature extraction multiple times, and that really just means the acquisition of the data from the digitized images. And really there are two primary ways to do this. The first is kind of this top arrow here, and I'll just call that the agnostic type, where you're using an approach that's agnostic to the image and extracting all of the data from that image, again, without any prior knowledge of what that image is, and using those features then to predict whatever, your outcome or test the association with, the outcome that you're interested in. The bottom one is where it takes into account the knowledge of the tissue and the cells that you have. And so that approach uses a means to identify nuclei, which are, kind of anchors that are used to identify cells. And then that approach extracts information about those cells once the cells have been identified. So features of the cell wall, the cytoplasm, and then the nucleus, and then those features are then used to construct additional models or a model of your outcome. So one is agnostic, and the other is done with the knowledge of the tissue that you're studying, that it is cellular. And so the example that I'm going to show today from our data is really using this agnostic deep feature extraction method. So yeah, like I said, I really have this love of underutilized data, and H&E slides definitely fit that description. However, the data is far from free, and the cost is really in the precious time of histotechnologists and pathologists, sort of listed or shown here on the left side, part of our multidisciplinary team to pull all of this off. They're the people who have cut, prepared, reviewed, and meticulously annotated thousands of H&E slides for this project. There's a lot of additional data to be mined, and really this wouldn't be possible without them. And then on the right-hand side, on the top, I have my collaborators within public health sciences, mentor of mine, Dr. Rabicki, who I continue to work with, Sudha Sadasavan, who's really manages this entire project. Ian Lovelace, one of our analysts who continues to work on this data. And then also our urologist colleagues, Dr. Craig Rogers and Faraz Abdullah, who really keep us laser focused on on the clinical questions that matter the most. So I just want to acknowledge that this work wouldn't be possible without all these people and more. So again, the application of digital pathology that I'm going to talk about is in the area of prostate cancer. And so prostate cancer is the most frequently diagnosed malignancy in men, and it accounts for about 14% of all malignant cancers diagnosed overall. African Americans suffer really a greater burden of this disease. They have almost twofold higher incidence or risk of developing prostate cancer in their lifetime relative to European Americans. And they also have a more aggressive disease phenotype. However, prostate cancer accounts for about 4.8% of all cancer-related deaths, indicating that the majority of men with prostate cancer will not die as a result of it and will rather die with it. So therefore, it's really important to identify early those who are at highest risk of aggressive prostate cancer to be targeted for early therapy. People always they think about that end of the spectrum, but then they also don't necessarily think about the other end of the spectrum, which are the majority of patients who maybe don't need aggressive therapy, or maybe can be sorted to can be spared an overabundance of treatment, and their disease can be followed up to determine whether they really need to have aggressive treatment. So it's important to have stratification models to indicate who's at highest risk and lowest risk of developing more aggressive or recurrent disease. So there are multiple risk stratification models that exist, and the Gleason grade group is an important component variable of all of them. As increasing group score is strongly associated with increased risk of disease recurrence. So the Gleason grade group is scored on a scale from 1 to 5. And it's really indicative of the differentiation of the tumor. So prostate cancer arises in prostate glands, and an increasing score reflects the increasing de-differentiation of the cells, which just means that it looks less and less like a normal prostate cancer gland. Again, increasing score here is associated with increasing risk of prostate cancer recurrence as well as mortality from prostate cancer. So the pathological Gleason groups are composed of 3 Gleason patterns: Gleason pattern 3, 4, and 5. So the Gleason pattern 3 on the left are really indicative of discrete, well-formed glands. So still looking like the definitely tumor, but more like standard glandular structure. In Gleason pattern 4, that structure starts to disappear with poorly formed or fused glands present. And then Gleason pattern 5 is really a complete lack of glandular structure with or without necrosis and with or without poorly formed or fused glands. So each tumor focus is assigned a group based on a primary and a secondary pattern score, where the primary is indicative of the pattern that dominates that foci of tumor, or has 50% or more of tumors of that specified Gleason pattern. So the group 1 is homogeneous for Gleason pattern 3. So call them 3 plus 3, where the primary score is first, the secondary score is second. And group 2 has Gleason pattern 3 and 4, but is predominated by Gleason pattern 3. Group 3 is also, it contains 3 and 4, but it's predominated by Gleason pattern 4, and the 3 being the minority. And then group 4 and group 5 having patterns of 4 and 5 present. So in group 4 being homogeneous for pattern 4. So while it's fairly clear what should be done clinically right now with grade group 1 and grade groups 4 and 5 tumors, there's really a lack of clarity for these intermediate grade tumors. So prior studies have shown that the percentage of Gleason pattern 4 within intermediate Gleason grade group 2 and 3 differentiates risk of recurrence within these grade groups. Again, grade group 2, for the grade group 4, for grade group 2, the grade pattern 4 ranges from 1% to 49%, given that the grade pattern 4 is like the secondary pattern. And because of these, the known differentiation within these by the percentage of Gleason pattern 4 present, it's been the recommendation of the International Society of Uropathologists to report the pathologist assessed, the human pathologist assess Gleason pattern 4 percentage in pathology reports. However, this is not commonly done, and even though they do differentiate within these recurrence risks, within these intermediate patterns, this percentage currently isn't included in recurrence risk prediction, and the question is why? And the answer is that an accurate and reproducible assessment of the percentage of tumor cells is really challenging for a human pathologist. And we and others have thought that this might be an area where an artificial intelligence approach might be able to aid the trained pathologists and then facilitate the incorporation of this information into recurrence risk predictors. However, yeah, and you know, another reason why this isn't incorporated is because many healthcare systems, they don't have trained and experienced uropathologists who are specialized in that area. And studies have shown that the reproducibility of the pathologist assessed Gleason group score is even lower among general pathologists relative to the specialized uropathologists. Again, we need, you know, some tools to aid in this standardization. And so this is sort of leads to our study questions. And so our first question is, you know, can we develop an artificial intelligence convolution neural network model to identify and quantify these 4 prostate cancer cell types: benign glands, the stroma, which is really sort of characterizing the that's made up of the fibroblast structure, the extracellular matrix, and also the immune infiltrate. And then Gleason pattern 3 and 4 tumor cells. And then second, if we can develop this model, can we apply the quantification of Gleason pattern 4 percentage to differentiate risk of recurrence within intermediate grade tumors, the grade group 2 and 3 tumors. And so here we're defining recurrence as biochemical recurrence, which we define as 2 consecutive rising prostate-specific antigen levels following the nadir after radical prostatectomy or surgical removal of the prostate. So this study is based on a retrospective cohort of African-American prostate cancer cases that were diagnosed and treated with radical prostatectomy at the Henry Ford Health System between 2000 and 2012. Over that time, there were 874 African-American prostate cancer diagnosed, primary prostate cancers. And on the right, I just have some very simple descriptive statistics comparing the African-American cases that make up this cohort to the European-American cases that were diagnosed at the same time. And so what you can see is in all of these categories, age, Gleason group, and also pathologic stage, that they're actually quite comparable at diagnosis. So again, these were all individuals who were treated with radical prostatectomy. And the other thing that I wanted to point out here is that the vast majority of the tumors are falling into these intermediate groups where there's a lack of clarification as to what exactly should be done with those particular patients in terms of treatment. So then after we enumerated the overall cohort, we then included additional inclusion-exclusion criteria, that there were no prior malignant cancer diagnoses, no prior treatment for prostate cancer. These patients had to have formalin-fixed paraffin-embedded tissue blocks available from our biorepository, and then they had to have a minimum of 3 PSA test results following their radical prostatectomy to assess biochemical recurrence. And so our total sample size at the end of all of that was 298 individuals. So the okay, so like I said, we had 2 questions. One was the convolution neural network model development that was focused on identifying or differentiating these 4 cell types. And the second one was applying that model to assess Gleason pattern 4 percentage in the intermediate tumors and assessing how they're associated with biochemical recurrence. So for the model development, the CNN model development for question 1, we had a discovery sample that was composed of 43 grade group 1 tumors because they were homogeneous for pattern 3, and 22 grade group 4 tumors that were homogeneous for pattern 4. And then we had the remaining 16 and 5 tumors respectively that were used for validation. And then when we applied the model to assess Gleason pattern 4 percentage, we had 151 grade group 2 tumors and 61 grade group 3 tumors. So the Gleason pattern CNN model construction really starts with the generation of whole slide images, an image for each tumor, which was then annotated by our uropathologists and broken down into these tiles or square tiles or patches using the quantitative pathology software called QPath. These patches, which were approximately 3,000 per cell type, were then used to train the convolution neural network to detect the 4 individual cell types, which we then validated using the similar set of patches from the independent set of grade group 1 and grade group 2 tumors that I showed on the last slide. So modern CNN convolution neural network models have a large number of parameters, over 1 million. And so overfitting can be a huge problem if you have a small number of training images, as we do in this study. Therefore, we utilize a particular convolution neural network called the Inception ResNet version 2 CNN model for feature extraction in this study. And that neural network has been trained on over 1 million images from the ImageNet database. When you take a trained neural network like this from, that was trained on one a set of images, and you port it over to apply to, another independent set of images, they call this transfer learning. And then what we did was extend the vanilla Inception ResNet 2 neural network. We extended it by adding this additional last output layer that was really the classification layer for these 4 different prostate cell types of interest. So the accuracy of the model is based on the classification of the individual patches. The model reached an accuracy of 100% after 15 epochs in the training data. But that's the training data. So when we actually apply the trained model to the validation data, the overall accuracy was lower, but still quite high at 87%. And the validation accuracy values by cell type were 80% for the grade pattern 4, 78% for the grade pattern 3, 93% for the benign glands, normal prostate glands, and 92% for the stroma. So to give you a sense of how the individual patch predictions are related to the individual image, we can map the individual patches back to their coordinates on the image and create heat maps based on the predicted probabilities of grade patterns 3 and grade pattern 4. So on the left, you have an image of a whole mount prostate specimen, and there are 2 areas of tumor that are labeled. One on the top labeled as T1, and a smaller segment on the bottom labeled as T2. So the T1 region is 4+3, so a grade group 2 lesion. And on the bottom, it's 4+4, 8, which is a grade group 4 lesion. And so then on the right, you can see the heat maps for the grade pattern 4 percentage on the top, and the grade pattern 4 on the top, and grade pattern 3 on the bottom, where increasing intensity of red is indicative of a higher predicted probability of grade patterns 4 and 3. So if you look at this, top T1 lesion, you can see up here that it's predominated by red pixels in the grade pattern 4 heat map, and less red pixels in the grade pattern 3 heat map, which is indicative that there's more grade pattern 4 than grade pattern 3 in this T1 lesion, which is consistent with what the pathologist had scored. And then if you look at this T2 region on the bottom, you can see here that it's almost exclusively grade pattern 4. And when you look down at the same region here, you don't see any grade pattern 3 pixels, which is, again, is consistent with the homogeneous grade pattern 4 call by the pathologist. So then what we can do is we can apply that model to the intermediate grade group 2 and 3 tumors, and use that to calculate the grade pattern 4 percentage, really as the number of patches that are called as grade pattern 4 within the pathologist marked tumor area, divided by the total number of patches that were either grade pattern 3 or 4 times 100%. And so then we just plotted those percentages here in the graph on the right-hand side. And what you can see is that there's a lower proportion, a lower percentage of grade pattern 4 in grade group 2 lesions versus grade group 3, which had higher, which again is consistent. So this is just showing consistency between the neural network model and what the pathologist had identified. So while the grade pattern 4 percentage was not significantly associated with risk within grade group 3, grade pattern 4 percentage did significantly differentiate risk within grade group 2 tumors. So here with this hazard ratio of 1.18, which is to say that each increase of 10% in the grade pattern 4 percentage was associated with a 1.18-fold increased risk of recurrence. And to show this visually, we split the grade group 2 tumors into low, medium, and high based on tertiles of the grade pattern 4 percentage. And we can see the separation in biochemical recurrence risk at 5 years with the biochemical recurrence risk-free probabilities of 80%, 73%, and 54% for the low, medium, and high percent GP4 groupings. So I'll also say that this estimate here is also adjusted for other predictors of biochemical recurrence, such as age, prostate specific antigen value at diagnosis, and tumor stage. So I think what I really wanted to show you was this, interesting new use of old data towards the production of a tool that could actually be used by pathologists in grading their prostate cancer. So we showed a high fidelity assessment of Gleason patterns using the convolution neural network. Also, this is an objective quantification of, Gleason pattern 4. So this is something that potentially could move towards clinical application, but we would need a whole heck of a lot of additional data to get there across multiple institutions, across multiple, with consensus panels of pathologists scoring these. Really, we had 2 pathologists that scored each image where they had consensus on the grade group. And then consistent with the literature, we showed that there's this differentiation of risk within grade group 2 tumors. And that you can have more refined risk assessment within intermediate grade tumors, where more aggressive tumors, might be sorted to actually treat them more aggressively. And the ones that are on the lower end, the Gleason grade group 2 tumors that are on the lower end of the spectrum might be sorted to surveillance as opposed to immediate treatment. So we're kind of in the last stages of publishing this paper. We just submitted our second revisions, minor revisions at Modern Pathology. So, and then this paper's, I put it up as a preprint as well on Research Square, which is sort of like bioRxiv. So where are we going with this? Really, I think we want to expand out the data set that we're looking at to further refine the models, but we're also really excited about pushing this into the biopsy space. So if we can assess within the biopsy space, then that's where we can really assess whether surgical or some other primary therapy can be delayed, and to reduce the amount of treatment, unnecessary treatment in these less aggressive tumors. So in the positive biopsy space, but then we also think that we may be able to push this back even further into the negative biopsy space to determine whether these deep features that we're extracting that are predictive of more aggressive phenotype, if there is any evidence that they're in they're evident before full-blown prostate cancer is evident. So they might be there, but in a more subtle form. And so we have a large benign biopsy cohort that Dr. Rybicki has developed, and we're currently assessing whether we can use this information that we're gathering from the tumors into that in the negative biopsy space as well. So again, I just want to acknowledge the team that I put up there, which is really the current team, and then also past members within public health sciences. Again, Dr. Yale Chen, he really initiated all of this research and did all the CNN model development, along with a really amazing undergraduate from Carnegie Mellon, Tiffany Nakai, and then again, other pathologists. You can never have enough pathologists. So yeah, I'll end there and take any questions if you have them.